Hi everyone. We'll continue with production of mature mRNA in eukaryotes. And unlike bacterial mRNAs, eukaryotic mRNAs are modified at both the 5' and 3' ends. We have seen this picture in the previous lecture as well. This is a eukaryotic cell. It has a true nucleus, cytoplasm separated from the nucleus. This is part of the DNA shown here. After transcription, the primary transcript in eukaryotes is not mature. It's premature messenger RNA or pre-mRNA. And the pre-mRNA has to be processed and converted into final mature mRNA, which will be exported to the cytoplasm for translation. We did talk about this when we were comparing prokaryotic and eukaryotic transcription in the previous lecture. In this lecture, we will see part of the RNA processing. That's how this pre-mRNA is processed into mature mRNA. So we'll see 5' and 3' modifications. That is the changes done at the 5' and 3' ends. Let's first begin with the 5' modification. What modification is made at the 5' end in eukaryotic pre-mRNAs? In one of the previous lectures, we have seen that eukaryotes have 3 RNA polymerases, and it's the RNA polymerase 2 that carries out the transcription for the protein coding genes and synthesizes mRNAs. So once RNA polymerase 2 has started with the transcription and has made about 20 to 30 nucleotides of the pre-mRNA, a capping enzyme adds guanine nucleotide, most commonly 7-methylguanosine, to the 5' end. So what is this 5' modification? That is modification at the 5' end in eukaryotic mRNAs. This 5' modification is actually called as capping. So eukaryotic mRNAs have a cap at the 5' end. What is this cap, first of all? And when is this cap added? So as we spoke about, when RNA pol 2 has made 20 to 30 nucleotides of pre-mRNA, a capping enzyme adds a guanine nucleotide. So this cap is basically a guanine nucleotide, a guanosine, and this cap is not added after this pre-mRNA is completely made. This cap is added when 25 to 30 nucleotides of pre-mRNA are made. This cap, which is actually a guanosine or a guanine nucleotide, is added to the 5' end. This is a methylated guanine and methylated at the 7th position. So this is 7-methylguanosine. And we will see in this lecture that initially only a guanine is added, which is later methylated. This addition of cap, it involves a very unusual 5' to 5' linkage rather than the usual 5' to 3' linkage. This process is called as 5' capping, and not just this cap guanine is methylated, the sugars of the next 2 nucleotides are also modified by methylation. We are talking about RNA, mRNA. RNA is ribonucleic acid. It has a ribose sugar. So the ribose sugar of the next 2 nucleotides, which are the first 2 nucleotides, are modified by methylation as well. What is the function of this cap? Why this cap is added? Why a guanine nucleotide is added when the pre-mRNA is made in eukaryotes? This 5' cap remains throughout the processing, and it's even present in the mature mRNA. So if we go back to this diagram, this cap is being added when the pre-mRNA was made, or when 25 to 30 nucleotides of pre-mRNA have been synthesized, but this cap remains in the final mature mRNA. It remains throughout the processing, and it is there in the final mature mRNA we can see even in the mature mRNA, which has been exported to the cytoplasm. So this cap remains throughout processing, present in the mature mRNA. What role does it play there? It protects the mRNA from the 5' end from degradation by exonucleases. Nucleases are the enzymes which can degrade nucleic acids. Here we are talking about mRNA, so we are talking about nucleases that can degrade RNA, RNases, ribonucleases. There are exonucleases, there are endonucleases. Exonucleases are the nucleases which can cleave the nucleic acid from the end, whereas endonucleases are the nucleases which can cleave the nucleic acid from somewhere in between. So exonucleases can easily degrade the mRNA from the end, but this cap protects the mRNA from degradation by exonucleases at the 5' end. So this cap ensures the protection at the 5' end, and this protection by the cap occurs due to the unusual 5 to 5 linkage. Is there any other function of this cap other than protection from exonucleases? Yes. The second function is this cap, as we have told, that it remains in the mature mRNA even when the RNA has been exported to the cytoplasm, and this cap also promotes translation. As we have seen in the previous lecture, that after the RNA processing is complete, the mRNA is exported to the cytoplasm for translation, and this cap is important for the binding of the ribosomes as an initial step of translation, and it promotes translation. Let's see the process of how this cap is added with the help of this diagram. This is a pre-mRNA of the eukaryotes. This is being synthesized. At the 5' end, there are 3 phosphates. One of the 3 phosphate groups at the 5' end is removed. Before the guanine cap is added, out of the 3 phosphates, one of the phosphate at the 5' end is removed or cleaved. And if one of the phosphate is removed or cleaved from the 5' end, out of 3, that leaves only 2 phosphates. Now the guanine nucleotide is added with its phosphate group. So guanine is being added here with its phosphate, so that makes again 3 phosphates. And the GTP molecule is hydrolyzed, that results in addition of the guanine. And we have told that the guanine is methylated at the 7th position. But at this step, you can see when the guanine is added, it's not methylated. After the addition of the guanine cap, methyl groups are added to the 7th position of the guanine, resulting in 7-methylguanosine, and the capping is completed. After that, the 2' position of the sugars in the 2nd and 3rd nucleotides are also methylated. The sugars, the ribose sugar in the 2nd and 3rd nucleotides are methylated, and the base on the initial nucleotide may also be methylated. So this is how modification takes place at the 5' end in the mRNA of eukaryotes. Is there a modification at the 3' end? Yes. And in this diagram, we have seen that there are string of adenine nucleotides at the 3' end, that is 3' modification. So let's see the 3' modification now. Most eukaryotic pre-messenger RNAs become modified at their 3' ends as well by the addition of a sequence of about 50 to 250 nucleotides. So eukaryotic pre-mRNAs are modified at their 3' ends also by addition of around 50 to 250 nucleotides. Which nucleotide? Adenine. Only one nucleotide is added in various copies, and that is adenine, resulting in a poly A tail. There is no DNA template for the poly A tail. So when the transcription is happening and the gene is transcribed into mRNA, the DNA sequence is used as a template, obviously, for the synthesis of mRNA, but there is no DNA template for this poly A tail. The information for this poly A tail is not in the DNA. So for example, if we say poly A tail is added, complementary, so there should be thymine nucleotides in the gene or DNA. There's no DNA template or no information in the DNA for the poly A tail. Poly A tail remains when the pre-mRNA is processed to the mature mRNA. So this poly A tail remains during the processing and it's there in the mature mRNA. mRNA molecules with 3' poly A tails are called poly A plus mRNAs. What is the function of poly A tails? If there are functions of the 5' cap, there will be some function of this poly A tail or 3' modification. We have seen and spoke about coupled transcription and translation in prokaryotes in bacteria in the previous lecture. And in case of eukaryotes, since the first transcript is not the final mRNA, it's pre-mRNA, and it has to be processed, what we are learning, RNA processing, and it has to be processed into a mature mRNA, and this transcription and processing takes place in the nucleus itself. But the mRNA in eukaryotes has to be transcribed and processed completely before it is translated, because the translation is going to happen in the cytoplasm. Ribosomes are in the cytoplasm, so mRNAs are exported from the nucleus to the cytoplasm, and poly A tail plays a very significant role in the efficient export of this mature mRNA from the nucleus to the cytoplasm, so which becomes the first function. That poly A tail is required for efficient export of the mRNA, mature mRNA, from the nucleus to the cytoplasm. Like 5' cap, the poly A tail also protects the mRNA against the degradation by the exonucleases. Yes, to protect the mRNA from degradation by exonucleases, there is a cap at the 5' end which protects the 5' end, but some protection is required at the 3' end also. Otherwise, the nucleases can degrade the mRNA from the 3' end if they cannot do it from 5' end. So poly A tail does the same or serves the same function that a cap does at 5' end, and poly A tail protects the 3' end of the mRNA from degradation by exonucleases. And there's a third function, that is promotion of translation. Poly A tail also plays important roles in the initiation of translation by the ribosomes and in processes that even regulate the stability of mRNA. So for efficient export from nucleus to cytoplasm, protection from degradation by exonucleases at the 3' end, promotion of translation, and to regulate the stability of mRNA, these are the functions of the poly A tail or the 3' modification. So in this lecture, we have seen the RNA processing in eukaryotes, that how a pre-mRNA is converted to mature mRNA, but we have seen only one half of the processing, that is modifications done at the 5' end, 5' cap, and at the 3' end, poly A tail. In the next lecture, which will be the last lecture of this course, we will see rest of the RNA processing and how the pre-mRNA finally gets converted into mature mRNA.